part of the chamber is open and part of it's closed. Of course, rats like to hide. Well, if you put them in the open one, normally they'll run into the closed section. If they're depressed by whatever mechanism, either endogenously or biochemical induction, they'll stay in the open area for longer. They like count how much time they spend in either area.
There's a variety of other tests they have. There's one called learned helplessness, where they create a situation where the animal just can't control their environment, so eventually they just stop trying. It is a learned phenomenon in the rat, anyway. Then there are other strains of rats that just kind of have low serotonin or altered serotonin and just don't interact with their environment, and so they're basically a good model, but you know, you can't ask them, so do you feel like committing suicide, you know? Do you have suicidal ideation? They just never come in and find a rat with their wrist, like, even if you put a razor blade in there, they just don't do it, you know? So there's unique human attributes to the disorders that discovered. I don't see anyone downtown right now, but I'll go ahead and so both microphones are on. Got the camera, and the teacher cam. Well, good afternoon, everyone. I feel like we should just be leaving, or actually be long gone, rather than just starting. But the world changed on us, so here we are. I'll switch over to the desktop now, and we'll begin with lecture 24, the urogenital system. And we start first with the discussion of the development of the urinary system and the adrenal gland, and then we'll proceed to the Wednesday and Friday lectures, lecture 25 and 26, on the development of the reproductive system, the genital part of the urogenital system. Again, we'll have the Friday lecture pre-recorded and posted, so we will not expect anyone to be coming to class. We'll have that posted as we did last week. That seemed to work out quite well. There will be a couple of lectures that Marco will do, probably later in the semester, very likely close to Thanksgiving and just after Thanksgiving. And again, that's part of the exercise that he's undergoing with me as kind of a trainee, I guess. And otherwise, all of the Friday lectures, there's only 5 left in the semester anyway, will be pre-recorded and posted because I have this conflict, as I had mentioned, with an off-campus activity at the same time, 2 to 3. This seems to work out well, and I appreciate any kind of feedback through the WebCT mailbox system, whether you like the way that it turns out, whether you like the PIP, where you see the human being giving the lecture versus just hearing the voice and seeing the arrow move and all that kind of stuff. We're continuing to develop things. Haven't received any feedback from anybody on exam 2. So it seems to be going well. Look, the grades are a pretty good distribution. No one seems to be in trouble. And we'll proceed to use that same model for exam 3, which will become available after class a week from today. Excuse me. The first 23 questions of exam 3 are from a slide set. I will pre-record the questions for those set of slides, and again, I will forward to you the paper copy, in other words, a Microsoft Word attachment that you can print out to answer those questions. The slideshow will be posted and made available to you on the same day, so you can decide how you want to interface with the slides, whether to print them and have them there to look at, following the arrow on the the streaming video, or you can have the slideshow loaded so that you have it on your screen, and then in the little window have the streaming video, and then the printed out copy of the answers, the possible answers, and you can record your answers that way, and then when you log on to start your exam on WebCT, you can just enter those responses, those selections in for the first 23 questions, and then proceed to answer questions 24 to 50. So exam 3 will be the last one with 50 questions. So remember that the final exam will be, again, online, the same mechanisms, but it'll have the 100 questions. And on your syllabus, you have the method available to work either with your percent grade or points accumulated to see where you stand along the way. So the urogenital system, we begin with a discussion of the urinary system and the adrenal glands. And of course, one of the easier approaches is to take a quick look at the adult urinary system, so we have that fresh in our mind as we proceed to look at the development. So the organs that we're talking about in this organ system are the kidneys, ureters, bladder, and urethra. And we start first with the question, the urogenital system develops from, and we will see it develops from the intermediate mesoderm. We know that there is some contribution for epithelial lining from the endoderm, but again, in this type of a question, we're looking for the best possible answer, and the majority of the urogenital system is going to come from the intermediate mesoderm. The epithelial lining, that contributed by the endoderm, is really minor compared to everything else, and we'll see that as we proceed. So the kidneys that form during intrauterine life in the human include 3. The earliest, the pronephros, the intermediate, the mesonephros, and then the ultimate, the metanephros. So we will follow the development of all 3 of these. The one that by far has remnants that persist in adulthood is going to be the metanephros, but there will be some mesonephros that persist as well. So that's kind of an example of how this style of question that has a question and all of these are correct. What I just stated is an example of how that can be converted into another question. So we're going to try to envision how the neonatal, adult, of course, kidney system develops, and we'll see that it actually develops here in the pelvic area, and migrates up into this posterior abdominal position to join with the adrenal gland that develops in this area as well. And today's lecture will cover a little bit of the adrenal gland development, and we'll see more of that as we proceed through our discussions. So the ureters develop from a structure, and then the urinary bladder will develop in that area, as well as the urethra. So for today, we'll just follow mostly the kidney, ureters, and the vasculature for the kidneys, and then we focus on the cloaca and all the other structures in that area for the development of the reproductive system. We'll see the final stages of development and the contributing factors for the urinary bladder and the urethra. So we're really going to spend our time today talking about the kidneys and just begin to introduce the adrenal glands. So early on, the nephrogenic ridges or cords are going to be bulging in the posterior abdominal wall. We saw that in cross-sections in the previous lecture. Actually, we saw the bulge created by the adrenal glands, and then one that I said that was the pancreas, which was totally wrong. It was these nephrogenic ridges, but here, retrospectively, I can beg your forgiveness. So the pronephros is the first to develop in this area of the nephrogenic ridges. And, of course, it's the intermediate mesoderm that's differentiating into actually all of these structures. And we'll see that in an illustration in a moment. So the pronephros are intermediate structures. They are transitory structures. They appear and then disappear. So no one knows why. Again, it has something to do with ontogeny, recapitulating phylogeny, but the contribution in the developing human embryo is not completely understood. They don't become functional, and they differentiate into cell clusters and tubes in week 4 to produce a pronephric duct. So they seem to get a good start and just kind of disappear and really don't provide much. And we see that illustrated here. On the right side, we can see the pronephros developing most rostrally or cranially, the mesonephros in the middle, and then the metanephros is going to develop off this little bud here that we see, the metanephric diverticulum, or it's called the ureteric bud. So the metanephric diverticulum reminds us that it's going to be the metanephros. So the mesonephric system is developing through this full length of the posterior abdominal wall, seems almost to extend up into the primordial thoracic region, but we know that the embryo is going to elongate quite a bit, and then ultimately, as a fetus, elongate even more. So things end up repositioned, basically. Of course, the pronephros will not persist whatsoever. But you already see that the metanephric diverticulum is way down here near the cloaca, so it's easy to see it's really in the pelvic region, and ultimately we'll see that it has to migrate rostrally or cranially to gain its final position. We'll follow the development of the mesonephros next, but we do see that there's a duct system that has become established, the mesonephric duct, and from that there are these little tubules that push off into this red-colored portion, which of course is intermediate mesoderm, the cells beginning to cluster and communicate and decide what they're going to do. So this mesonephros or mesonephri are caudal to the pronephros. They develop late in week 4. They do function as interim kidneys. So the fluid balance within the embryo and associated, as we said, with which fluid? The amniotic fluid came there eventually. Is very balanced. There's a recycling of everything. So a swallowing when everything is patent, the gut tube is patent. There's a swallowing of the amniotic fluid, an absorption, a filtration by the kidneys, and then the excretion back into that. So there is that complete recycling, as well as any diffusion of fluid into the embryo through the diffusion mechanisms that we've talked about from the very beginning, that we know as the embryo gets bigger and bigger, diffusion is going to be the least likely route for any liquid to get in or out of the embryo, and then it's going to become the pathway through the respiratory and the digestive system that any liquid would get into and out of, urinary system certainly out of, for the amniotic fluid. So the mesonephros will be the first renal system that will come into play for filtration of blood and elimination, excretion of waste products and liquid into the amniotic fluid. So we will follow the development of functional glomeruli and tubules. This will degenerate by week 12, so we basically have an 8-week time frame across which these develop, function, and disappear. So there's a number of tubules that are emptying into that one mesonephric duct. And then some of the components are going to persist as the male reproductive structures. So we don't lose all of these structures entirely, so there isn't a wasted effort of the embryonic development. So which parts of the mesonephros persist in the adult? And let's list their derivatives for male and female. So the excretory tubules of the mesonephros, the mesonephric ducts that we just looked at, will become the mesonephric or the Wolffian duct in the reproductive or the genital part of urogenital system. So in the male, these mesonephric ducts or the Wolffian ducts will persist as the ductili efferentes of the testes, the ductus epididymis, the ductus deferens, the seminal vesicle, and the ejaculatory duct. So obviously several structures in the male will be produced by these persistent mesonephric ducts. In the female, they're just these vestigial structures, the epoutheron and the peroupheron, and we will wait until we study the female reproductive system before we see these. So they really don't contribute anything functional to the human female reproductive system, but there are some remnant vestiges that do persist. In both sexes, that contributes to the trigone of the urinary bladder that was mentioned in the previous lecture, that last slide. The trigone is the spot where the two ureters come in and the urethra exits from the urinary bladder, and a ureteric bud, the Kupfer's duct, that we'll see a bit later. So some parts are common to both male and female for the urinary system, but for the reproductive system, by far the mesonephric ducts contribute mostly to the male reproductive tubular or duct-like structures. So from these nephrogenic cords, nephrogenic ridges or cords, we will see the metanephros develop as well, and metanephra is the plural. So there will be permanent kidneys that will form from these. And then about week 5, they start to kick in, and by week 9, they're functional. So we can see that the metanephros have a chance to develop and become functional, contribute to the filtration process, just as the mesonephros are tapering in their functional contribution, and they disappear. So let's take a look at these in these illustrations from the textbook. So first we see the cloaca and the promesonephric duct, the mesonephric duct is draining into there. By this time we have the rudimentary pronephros, and we recall the nephrogenic duct and the tubules within the nephrogenic cords that are forming. So we'll zoom in on that with cross sections at the level shown here for panel B and see what we have. So we see the nephrogenic cord forming here, and then off to the side that we can't really perceive from this lateral view of the embryo, we now see that the mesonephric duct and the nephrogenic cord are actually physically separated. So they will have to meet with each other at some point. We see the lateral folds closing in on the digestive system with the yolk sac pushing into the umbilical cord. We remind ourselves of the dorsal mesentery, the dorsal aorta, and the neural tube and the neural crest cells in this area as well. We now zoom in just to see the level of the dorsal aorta. A branch would be going to the dorsal mesentery to the gut tube, as we recall. So the visceral peritoneum onto the mesentery, the dorsal mesentery reflecting off onto the parietal peritoneum, and we see the urinary system forming outside of the parietal peritoneum from the very beginning, so it is primarily retroperitoneal. The mesenteric structures of the gut system, the gut tube system, that started with the dorsal mesentery and lost it, like the ascending and the descending colon, they end up secondarily retroperitoneal because they were initially mesenteric and then they lost their mesentery, so they're secondarily retroperitoneal. The urinary system, as we see very nicely here, never had a mesentery, were outside the peritoneum from the very beginning, so they're called primarily retroperitoneal. Well, now we see that the mesonephric vesicle has elongated and especially pushed laterally to meet the mesonephric duct. They make contact, and now it begins to form the mesonephric tubule. So we recall that the adult kidney has the glomerulus, which is the capillary tuft. That's the vascular side of it, along with a variety of other blood vessels that we'll see developing in a while. And then the tubule that has the duct systems within it, or makes connection with the duct system that leads into the urinary bladder. So far we're just seeing the initial development of the tubule and the duct system. We have yet to see the vasculature, and this slide, of course, introduces that aspect of the development. So obviously from the dorsal aorta, there will be a segmental artery that would have basically pre-existed, that would come into this area, or where it is in this area, or infiltrates into this area. It's stimulated to produce a capillary tuft. So we would have the afferent arteriole, as we know it to be called, the glomerulus, and then the efferent arteriole. And the glomerulus will form an association with a portion of the mesonephric tubule. And then on panel E, we see that it's going to, that portion that associated with the glomerulus will differentiate into the Bowman's capsule, so that we now have the renal corpuscle. The renal corpuscle is the functional term for the association between the tuft of capillaries and the glomerular, the Bowman's capsule that forms a layer on it. So we now think of Bowman's capsule as a balloon, and a tuft of capillaries pushes into that balloon. Whenever we talked about anything with this metaphor, we always said we end up with a visceral layer. So there's a visceral layer of Bowman's capsule on the capillaries. And then reflecting off from that is the parietal layer of Bowman's capsule. Between the visceral and the parietal layers is Bowman's space, and the filtrate passes from the capillary through the visceral layer of Bowman's capsule into the space, and we now have the filtrate that passes through the tubule for reabsorption and additional secretion from the tubule cells to produce the urine that would be in the mesonephric duct and exit on down to the cloaca, as we had seen. So we see that the afferent arteriole contributes to the formation of the glomerulus. It coalesces into the single vessel that leaves the efferent arteriole that forms a second capillary bed surrounding the tubules for reabsorption or action for additional secretion. And then it coalesces into the venous drainage of the kidney going into the posterior cardinal vein. So this is the formation of the functional mesonephris. And we see on the medial, the ventromedial surface of this ridge, the formation or the primordium of the gonad, or the testes or the ovary, depending upon the genotype of the embryo. So ultimately, this is going to become associated with the mesonephric duct, especially in the male. But for now, we have a functional filtration and excretory system. So now let's take a look at the formation of the permanent kidneys. Obviously, we're going to see something similar to this for the tubule formation and meeting with some mesenchyme that's going to differentiate into the glomerulus of the metanephros and the formation of the final filtration and tubular system for the permanent kidney. So we'll take a look at the renal cortex formation. We know that that's where most of the filtration occurs, and then the major ducts are going to be in the medulla. So the metanephric mass of mesoderm is going to be present to meet with the invading metanephric diverticulum or ureteric bud, and they will interact with each other similar to what we just saw to form the tubule system, the glomerular system, and form that final structure. So the induction is caused by the differentiation of the invading and growing collecting tubules, part of that ureteric bud or mesonephric diverticulum that becomes the ureteric bud. So it forms the components of the nephron, the glomerulus, Bowman's capsule, proximal tubule, loop of Henle, and the distal tubule that we all know. So the collecting duct and the collecting tubules are going to be from the ureteric bud, and the rest of the nephron, glomerulus, Bowman's capsule, proximal tubule, loop of Henle, and the distal tubule, epithelium, connective tissue, vasculature is all going to come from mesenchyme in these ridges. So the glomeruli start to develop in week 10, just as the mesonephros is beginning to disappear. So the maximal number of glomeruli will have developed by week 32, and we know that there will be nearly 1 million per kidney. So there's a tremendous amount of activity that's occurring here. So this illustration lets us vision this again. So the mesonephros is present through the full length of the dorsal body wall, and we talked how that develops. And we see now the mesonephric diverticulum, or the ureteric bud, pushes into this area, and we see the metanephric mass of intermediate mesoderm that it encounters. They begin to talk to each other and trigger developmental events to form the permanent kidney. So let's follow that in this illustration. So very early on, the ureteric bud pushes into this metanephric mass, begins to split and forms the renal pelvis, the connection. It becomes the ureter. It's obviously going to elongate over time, as will everything else. So the renal pelvis will lead at this point to this bifurcation, forming the major calyx. Then we will have multiple major calyces, at least 3 as we see here, and they split. So again, it's this outgrowth of a tubular structure, divides. Same thing we saw of the biliary tree for the liver and the air passageways, the bronchi, the trachea, bronchi, bronchioles for the respiratory system. So it continues to divide and pushes into this mesenchymal cluster of cells, the metanephric mass of mesoderm, and triggers clustering of these cells. There will be a metanephric mass taking a crown-shaped appearance close to the surface, obviously in the cortex. Slightly deeper will be the mesenchymal cell clusters. So we're seeing the primordia of the superficial and the deep glomeruli that will begin to form, and their associated tubules will also develop. So the original ureteric bud is growing and branching, and it will stop at one point where we see here this arched collecting tubule, and then they have this straight collecting tubule, and then there will even be grooves between the lobes that are forming. So the embryonic and fetal human kidney is lobulated, but by birth and very shortly after, it will no longer be lobulated. The adult kidney is not lobulated. Even though there are lobules, you don't see the little bulges on the surface. If you've ever been in a store and you look at the bovine or the cow kidneys that they sell there, they're still lobulated in the adult form. So you see these little bulges on the surface. So initially they're lobulated in humans, and then later that disappears. I want to add one more point before leaving this slide, we see the straight collecting tubule and the arching collecting tubule. I will now refer to the straight one as the collecting duct, and then these arching ones are the ones that feed into the collecting duct as the collecting tubules. And the collecting tubules, we will see, will become continuous with the rest of the nephron, the distal convoluted tubule. So that's why we'll use the term tubule for that portion, but then this major one leading into the minor calyces, or that primary one, the straight one, we'll call the duct, because it's quite a bit bigger than the tubules leading into it. So this illustration now will let us take a look at and follow the interaction between these arched collecting tubules and the nearby mesoderm or mesenchyme and follow the development of the different glomeruli. So we see that the metanephric mass of mesoderm arches around, and there will be a clustering of cells. And as well, deeper in, we saw the metanephric vessel forming. Both of these will become associated with one of these arched collecting tubules. And once there's communication, then on the more superficial areas, we get a metanephric vesicle forming. And where there was already a metanephric vesicle deeper in, it'll make contact with the arching tubules, and we'll see the differentiation of the metanephric tubule. And there's still mesenchyme in the surrounding areas. We have to form everything else, the connective tissue, the vasculature, and all of that. So we keep that in mind. We also are reminded that neural crest cells will have undergone some differentiation and sent sensory processes into this area. We know that there's a neural sensory system as well. There will be postganglionic sympathetic axons. Again, the postganglionic sympathetic neurons in their entirety, dendrites, cell body or soma, and axon, originate from neural crest cells. So there's tremendous neural crest cell contribution to this area for both sensory and motor functions. We know the sympathetic nervous system very potently and powerfully controls the blood flow through the glomerulus. So when we see vascular, we think neurovascular and lymphatics at the same time, all the time, every time. So starting in panel C over here on the left, we begin to see that the deeper structures begin to differentiate first. So the nearby mesenchyme will start to form the arterial side of things, and these are segmental branches coming off the dorsal aorta. They'll form the renal artery. They will have formed as branches of the renal artery. And what we will see is that there will be multiple renal arteries that will give arterial supply to the metanephros, and basically it will take on new ones and abandon old ones as it migrates up through, cranially, up through the posterior abdominal cavity. So it's really quite an amazing thing that it can be accomplished at all. At this stage, we are seeing the very early glomeruli forming deeper, and then they progress toward the more superficial levels over time. But it's the same process. We begin to see the interaction of the vasculature, primordial mesenchyme with the this remote portion with reference to where the urine would actually be formed ultimately. So I call it remote. And we see the formation of the renal corpuscle. Let's just use that term. So Bowman's capsule is formed over the glomerulus. And then the tubule closest to that will become the proximal convoluted tubule. Then there's going to be the loop of Henle developing in between as this whole tubule begins to elongate. And then the farthest from the glomerulus is going to be the distal convoluted tubule that connects with the collecting tubules, and multiple collecting tubules empty into this straight collecting tubule that we refer to as the collecting duct. The collecting duct then opens into the minor calyces that leads into major calyces that lead into the pelvis of the ureter that lead into the ureter. The ureter drains into the urinary bladder. So we now see in our continued discussion of the formation of the permanent kidneys, the renal medulla. So it arises from the ureteric bud because the renal medulla is primarily collecting ducts and collecting tubules. But we know that there are other cortical tissues that form between the pyramids. I'm not going to get into that detail at this time. But basically, the renal medulla contains collecting tubules and collecting ducts, or if you want to stick with our present terminology, the arching tubules and the collecting tubules and the straight collecting tubules are what we find in the renal medulla. So by definition, then, of content, it is formed by the ureteric bud or the metanephric diverticulum, and it is at the distal end of the mesonephric duct. So it forms the renal pelvis as well, as we saw, the renal calyces, major and minor, and the collecting tubules or collecting ducts and the collecting tubules by my terminology. And it has induced a branch. It was induced to branch by the presence of the developing nephrons. So that interaction with the metanephric vesicle triggered the whole series of events. And then the ureter arises from the mesonephric duct because the the ureteric bud was a diverticulum from that original mesonephric duct down near the cloaca. So from which sources does the kidney develop? List the structural elements of the kidney developing from them, and then from which structure does the ureter develop? So the metanephric mesoderm produces the nephron. The ureteric bud, as we said, contributes the collecting system, the collecting tubule and the collecting duct portion of it. And then the ureteric bud is what gives rise to the ureter. So what is the origin of the ureteric bud or Kupfer's duct? And, of course, that leads to the next part of this question. What are the derivatives of the ureteric bud? So the ureteric bud, develops from the dorsal wall of the mesonephric duct. We made that point before. And then the rest of it persists as the Wolffian duct to contribute to the male reproductive duct system if it develops along the line of the male, especially in the genotypic male that will occur normally. Its derivatives, the ureter, renal pelvis, major and minor calyces, papillary ducts, and the collecting ducts. And those are even two additional terms. The papillary ducts are what I call the collecting ducts. What they're calling the collecting ducts would really be the collecting tubules by our terminology. So more than enough names for the same things. Thank you very much. So the migration of the kidneys. So we mentioned this early on. We had 3 sets of kidneys that develop in the embryo, pro, meso, and meta, and they develop in that order from cranial to caudal. The caudal one is the one that persists, and we know that it ends up moving out of the pelvis toward the cranial end of the abdominal cavity. Of course, it doesn't get into the thoracic cavity, but it ends up starts in the caudal-most part of the abdominal cavity and ends up in the cranial-most part, so we know there has to be a migration. So these kidneys originally form the pelvis, ventral to the sacrum. As the embryo lengthens, the kidneys ascend. So it's kind of an ascension, but it's really kind of well, I'm fixed here, and as you move away, then I have to reestablish new vascular connections, and since the urinary bladder is going to be drawn away from me, the ureter is going to have to extend. So there's always this relative kind of a thing in the embryo. Maybe it does ascend a little bit, but not like we picture the distance in the human, we're way down here in the pelvis, and then this metanephric kidney has got to move all the way up into the uppermost posterior abdominal wall. It's a shorter distance, and things just reposition a little bit and get longer as the embryo lengthens. But this does occur until the kidneys hit the adrenal glands. Once they communicate with each other, then there's no more movement that occurs. So the hilus of the kidney, that point of an organ where structures enter and exit, is called the hilus of the lungs, hilus of the kidneys, hilus of the liver. So this term is used for each organ. It will rotate from an initial anterior or ventral position toward a more medial type of adult position. The blood supply was originally derived from the common iliac vessels down in the pelvic region, and it changes as the kidney migrates cranially to the adult branches from the abdominal aorta. And we'll see an illustration of that in a moment. So how does the trigone of the urinary bladder develop? So the caudal portion of the mesonephric duct will gradually become incorporated into the wall of the urinary bladder forming in this area, and it will be at the point of the formation of the junction of the ureters and the exit of the urethra, and therefore it is trigone. It has three corners. And later the mesodermal lining of the trigone will be placed by the endodermal epithelium. So there can be defects, as in any case. So the blood supply defects are the most common. As we said, it's kind of extraordinary that we start with a low arterial supply to the kidneys, and then it grabs new ones as it ends up being repositioned higher in the abdominal cavity. Another possibility is the complete failure of generation of any adult or final or permanent kidneys, so renal agenesis. 1 in 1,000 is the occurrence. It's more frequent in males and females. Usually the left will be the one that's absent unless there is a bilateral renal agenesis, and it is caused by failure of a metanephric diverticulum to form in the first place, so it does not transduce the mesenchyme in that area to form the metanephric mesenchyme. There can be a malrotation or ectopic positioning from what is the final expected one, and a relatively common 1 in 500 cases, there will be a horseshoe kidney. So for some reason the inferior poles of the kidneys fused in the midline, so they get hooked on the blood vessels, the inferior mesenteric artery actually, so it does not migrate up. So it keeps its original arterial supply from the iliac vessels, and it does take on a horseshoe shape, and it stays actually inferior to the umbilicus and doesn't ascend. So there's no problem with this, and the only unfortunate thing is that there is an increased risk of a certain type of renal tumor called Wilms tumor. So developmental anomalies will quite often be associated with tumor development. So here in panel A, we see the case where 3 of these segmental arteries maintain their connection to the permanent kidney. So there's 3 renal arteries. This obviously is very important to anybody going in to do any type of surgery. And we see the suprarenal gland sitting on top of it and the ureter posterior to the renal arteries in this case. And then we see another example in panel B where it kept one of the earlier ones and took on the new final one. But this earlier one is actually causing a constriction of the ureter, and there will be an enlarged renal pelvis. Anytime you don't get the appropriate removal of the urine, you can get backing up all the way to the glomerulus, and you can get a cystic kidney. Definitely problems. In panel C, we see a case where there's 2 renal veins, In panel D, we've seen another example of left renal vein kind of forming this hugging arms around the aorta. So what is the developmental abnormality that leads to the malformation called the congenital cystic kidney? So if the collecting duct and the excretory tubules fail to join, then you'll have accumulation of urine in the convoluted tubules, and this will result in a cyst formation. So this is not from a backing up of urine. This is from a case of the formation of filtrate in urine, but the distal convoluted tubule doesn't end up connecting to those arching tubules, so you just have this cyst that forms with urine in it, with no place for it to go. And now we can just spend the last couple of minutes talking about the development of the adrenal glands. So we know the adult adrenal gland has a cortex and a medulla. The cortex will form of a mesodermal or mesenchymal origin. About week 6 it will begin to develop. It'll differentiate into zones later in the fetal life. The zona reticularis, the deepest one, will develop postnatally, and we'll see more details of this as we go along. The adrenal medulla, as we know, is of neural crest origin, so we will end up looking at slides where we see the intermediate mesoderm as mesenchyme begin to differentiate into the adrenal cortex, but it'll also be encountered by invading neural crest cells. The neural crest cells will develop into the adrenal medulla, and the mesenchymal cells into the neural crest cells. So just quickly, obviously, developmental anomalies can occur. So there can be congenital adrenal hyperplasia, or CAH. This is an autosomal recessive genetic disorder, and there ends up being an increased number of adrenal cortical cells and an associated or concomitant or resultant increased production of androgens. Remember, the adrenal cortex produces mineralocorticoids, glucocorticoids, and gonadal corticoids. So the androgens are one of the major ones, and there will be masculinization of females in utero. So this is what I said before. I said if the reproductive system develops along the male lineage. And I said this certainly will happen in the genotypic male. We now see a case where a genotypic female with congenital adrenal hyperplasia will develop along the lineage of the male reproductive system. So we will introduce the idea in the next lecture of the developing reproductive system. If nothing else triggers it, in other words, if testosterone doesn't appear in a high enough concentration, all embryonic reproductive systems will develop along the lineage of the female. So we use the term default. The default reproductive developmental process or lineage is female. So testosterone must be present to trigger male development. As we will see, if something interferes with that, it'll follow the default lineage and develop along the female directions. So obviously, there will be an increased production of ACTH in both the male and female that has resulted in this adrenal hyperplasia. So I appreciate your time and attention. Again, we will have lecture on Wednesday here in this classroom. I'll be here again. And then the Friday lecture will be posted online for you to view either at that time or another time. Now, last week we said that Marco would be here. This week we're not expecting anyone to come, so we're not planning to man this room or even arrange to have it opened. You will, if you wanted to view things at this regular scheduled class time, you'll have to go to one of the regular computer classrooms. So I might as well.